Hello, my name is Katrina Meyer. I'm a biophysicist working at the University of Zurich in Switzerland. So I was only 2 years old at that point, but I was definitely inside the wall in West Berlin, and I guess I was probably sleeping. I actually think I wanted to become an ice cream vendor, probably to be able to eat ice cream every day. So as you may have noticed, this didn't quite work out, but it wasn't maybe also the best idea of all times. My research breaks the wall of disease mutations. With the advance in DNA sequencing techniques, we are learning more and more about changes to our genomes. And today there are more than 100,000 known disease mutations in our DNA. But the vast majority of those has actually no known underlying molecular mechanism, which makes drug targeting of these diseases really difficult. So our solution is that we are trying to address this with a scalable screening approach to figure out the molecular underlying mechanisms. We have developed a first-of-its-kind screening method to study a very specific set of disease mutations and their molecular mechanism. And in this method, we can compare the interaction partners of a healthy protein and how that changes when the protein has a disease mutation. So this can be compared to a person's social media network and how this changes after some life-changing event, for example. And a very exciting aspect in this project was that we started following up on one specific mutation from a big database, and we did cellular and molecular biology techniques to study it in more detail. And then we actually also got in contact with the patient having that mutation, suffering from a specific form of epilepsy. And we were able to figure out that in her case, the protein is actually functional, but it is transported to a wrong position inside the cell, so it doesn't function just because it's in the wrong place. So with this we were able to solve her case and why she has this disease. So understanding the molecular mechanisms behind disease allows for development of targeted treatments. So one day this will allow for personalized medicine so that every single patient will get the treatment that is right for their specific mutation. We're thinking a lot about how to further increase the throughput of our method. Now we're in the hundreds, and to reach this 100,000 marks we have to get to the thousands. Also, like every other screening approach, our approach just gives the first hint on the molecular mechanisms, and that then need to be followed up on. So there are a lot of interesting clues lying around there that need further experiments. I have to admit that what keeps me lying awake at night are the small details of everyday work in the lab, especially when I do experiments that are supposed to be so easy and should just work, and then I'm doing them again and again and they just won't work. This really keeps me lying awake at night.